Hi everyone, today I want to share with you how to manage mouse lines with infertility. In a previous episode we covered the basics of how to manage a colony and discuss the common mistakes. In this episode I want to share what you need to know if your lines are sterile. Understanding what can go wrong will allow you to take the necessary steps to avoid delaying your research for months, if not years. All of this is from a first-hand experience because I have been studying mammalian infertility for over a decade now. I hope that what you learn in the next few minutes will help you avoid wasting resources. We will cover what needs to be done when you delete a gene that makes your males or females sterile. We will explain how to handle colonies that convert Flox alleles in the germ cells. And finally, we will cover the special case of genes in the X-chromosome required for male fertility. Before that, let's briefly recap the basic types of alleles we will be working with today. So today we will cover 4 allele types. The first one is the wild type. In this case, we have our standard genomic architecture with one exon followed by another one, and so on and so forth. These ones we are referring as plus in general. Then we will be working with Flox alleles that are very similar to the wild type alleles. The difference is that we will be inserting a LOXP site between flanking a critical exon. For example here, maybe exon 2. This one we are referring them as the FL alleles. The third type of allele is that comes from depleting or deleting this exon in the Flox, and that gives us the null that we symbolize as a minus, and at the end of the day it's just this exon followed by the LOXP allele. We deplete everything in between and we continue with the following exon, and this will generate an unfunctional protein that will give the depletion of the protein itself. The last allele that we are going to cover are the Cre alleles, that we refer to them as Cre positive, where you have somewhere a Cre recombinase expressed under a given promoter that will generate the Cre and will guide the transition from this Flox allele to this null allele. So these are the 4 alleles that we are going to cover today. Okay, let's start with the most basic situations regarding infertility in null animals. This section will help us revise the pedigree notation and will be critical for you to understand how to handle the Flox colony where the Cre is expressed in the germline. Here we will be covering infertile null animals. So imagine you want to expand a null colony. You can cross 2 heterozygous animals, and this is the notation in the pedigree. We are not going to write here the gene because we will always be working with the same, so we'll have a wild type and a null allele for this male represented in the square, and in the circle the female, again heterozygous with a wild type copy and a null copy. This can give us different combinations. So we can have like the wild type, we can have the heterozygous again, and we have also the null homozygous. Other possibility to get the null is to use null animals themselves, like we can have a male null cross with a female heterozygous. In this case we will have 2 types of possible alleles or combinations of alleles, like the heterozygous and the null. And this is a null male, but it can happen that these null animals may be sterile, so you won't be able to use this type of breeding in these situations. So the question now is, what happens if instead of having the null we have a situation where we have a Flox Cre and we are mating with a Flox animal in order to get again this Flox Cre positive or Flox plus animal. So imagine that these animals are sterile in homozygosity, the null. Can we do this sort of mating? So if the animals are not sterile, this sort of breeding will work. This one as well, because this would not be sterile, and this one as well, because there will be no problems there. If the females happen to be sterile, this breeding can be used because here the female is in heterozygosity. This one as well because the male is in homozygosity but the female is in heterozygosity. And this one is the same, so you can always use that. If the male is sterile, you can use this one because the male is in heterozygosity here. But the thing is, you cannot use this one. And in this case, whether you can use this sort of scheme will depend on the type of Cre. And this is what we will cover in a minute. Try to understand when this is possible. In this case, we'll have to keep the Cre allele in only one of the sexes. Let me show you what I mean with this. So let's talk about Flox Alleles and sex-specific Cres. So let's go back to the example where we have a male that can be sterile in the null situation, but that has here, is homozygous here for the Flox allele and has a Cre recombinase and is mated to a female that is wild-type Flox. So in this case, if the male is sterile with the null and the Cre is expressed in the male germline, this animal will be sterile, so we will not have this breeding. For the sake of this example, let's imagine that this Cre is actually expressed only in the female germline, so it wouldn't be expressed in the male. And what we want to do is to study the infertility of the females. So with this breeding, we will end up with a situation like this, for example, where we have a male that can be Flox-Flox and Cre positive, and a female that can be Flox-Flox and Cre positive as well. However, since the Cre is expressed in the female germline, this animal will be some sort of a chimera because it will have two types of alleles in the locus of interest. Where the Cre is inactive, that it will be Flox-Flox, and where the Cre is active, that these two Flox will become converted into the null. So what happens if we cross this female? Actually, this is the experiment that we want to do in some way. We want to check if these females are fertile when the allele is removed in the female germline. What we can get is a situation where we have the null allele and the Flox coming from this animal, and the Cre if you want as well. And for the male, we have the null, the Flox also, and the Cre positive. So note that when we use the Cre that is expressed in the female germline, whenever we cross a female that is Flox-Flox, the progeny will receive this allele converted into the null allele. So we cannot use this female for expanding the colony, so it can only be used to ask the question whether the female is sterile or not. So note that the genotype of the progeny will depend on where the Cre is expressed. So for example, if we have the Cre expressed in the female germline, as we had been assuming here, female germline, we will have here the conversion of these alleles. And note that none of these would be a problem if we have the Cre expressed, for example, in somatic tissue. So imagine that this is expressed in somatic tissue and this conversion only occurs outside the germline. The progeny would not care about where this thing is expressed, because ultimately the germline will transmit the Flox allele and they will be Flox-Flox Cre and Flox-Flox Cre positive, for example, if this is expressed in a somatic line. So this animal, if this is expressed in a somatic line, could be used not only for experiment but also for propagating the colony. And that's the main drawback or consideration that you have to do when you are expanding a colony that converts the Flox allele in the germline. Now, if we want to study early germ cell development using a Cre expressed in both sexes, we have some alternatives. Either we can use heterozygous animals or we can use an inducible Cre. Let me show what I mean with this. So let's cover now the example where we have Flox alleles and germline Cres that are expressed in both the male and the female germline simultaneously. So one breeding that we can use to test this is having, for example, a male Flox minus crossing to a female minus plus that has the germline Cre. This can give us a control situation where we have in the soma heterozygous for the null and the Cre, and in the germ cells this wouldn't change because there is not a Flox allele. These germ cells, and this would be the control. In the experimental we have the minus Flox Cre positive. Here the Flox and the wild type are playing the same function. In the germ cell, however, you have the conversion of the Cre, and you end up with the homozygous null if you have the Cre there. So these are the animals, and in case of the experimental we have like this chimera where you have the depletion in the germ cells. So this strategy can be a little cumbersome and take a lot to get there. There is a way, however, to use inducible Cres. So let me show you how this works. So you will have a male, for example, that is Flox cross to a female Flox, and we have a Cre that is inducible. Here usually is the ERT2-Cre that is used. And then you will have again a similar situation. You have the controls where you have the Flox alleles in the soma, and in the germ cells you have Flox as well. And for example, in the experimental you will have in the soma Flox and the Cre plus ERT2. And if you don't activate the Cre, you will still have this same genotype in the germline, ERT2-Cre, and only you will have the conversion of the Cre of the Flox allele if you have the Cre when you administer tamoxifen. So in the case of the control there will be no conversion because there is no Cre, and you will have in the germline this conversion to the null allele when you activate the ERT2-Cre by adding this compound known as tamoxifen. So these are again our animals, and we have a situation here where we have this genotype over here before the Cre and this genotype over here in the germline after the Cre. So after tamoxifen that activates the Cre, we have the conversion of these alleles and we have the null allele over there. This will give us the experimental animals. But before the Cre or before tamoxifen, you will have still the allele that would not be converted, and therefore you can keep the progeny of this breeding also for more breedings. Before we finish, I want to cover a special case where a gene in the X chromosome is required for male fertility. Because males have only one X chromosome, the allele can only be transmitted through the female in this case. So let's talk about this case where we have this X-linked gene required for male fertility. So in the case that you have the null allele in the male and you're crossing it with whatever female, so by definition this mating will not work. So you will have to transmit the allele always through the female. So the male will be plus on the Y chromosome and the female will have the null allele. And this will give us 4 possible scenarios. So the males can have the null and the plus in the X chromosome, while the females will always inherit a plus from the father and can be plus minus or plus plus. So this brings us the question, how can we generate double null females, or how can we test whether the females are sterile? I leave you with that exercise. I'll give you the answer at the end of the video if you can come up with a strategy to answer how to test for the female infertility in this land. Alright, that's it for today. We covered the restrictions for expanding a colony with male or female infertility. We discussed what you need to do to preserve the Flox allele when you are using germ cell Cres, and finally we got the special case of genes in the X chromosome that are required for male fertility. Hey, if you like this video, please subscribe to the channel. It is the best way to make this content available to people like you, including yourself in the future. If you want to learn more about mouse colony management, check the playlist now on screen, and I will see you there. So going back to the strategy of how to test for female infertility on a gene that is in the X chromosome and makes the male sterile. So we can come up with a strategy like this, where we have the Flox allele here in the X chromosome, the Y, and then a Cre that is specifically expressed in the female germline, and we can cross this to a Flox female, and we may end up with a situation of control experiment where we have the female Flox as a control and the female Flox Cre as the experimental. So that's the strategy. I hope you enjoyed the video, and I will see you in the next one.